tonight I'd like to welcome you to the last Science Cafe for 2020. We will be starting up again in January, but we're very happy to have finished on a high note. We've got Dr. Allison Gardner with us. She is a medical entomologist at the School of Biology and Ecology at the University of Maine. You know, she's talking to something tonight as Mainers or New England or a number of other places that is very pertinent to us, which is the black-legged tick, which is also known as the deer tick. Her work focuses on the ecology of infectious diseases that are transmitted by arthropod vectors. So that's things such as mosquitoes and ticks, and obviously ticks is the one we're most interested in because this season is actually one of the most active seasons for the adult ticks. And if you're anything like my family, we've been spending a lot more time outside than we would usually at this time of year. As it starts to get colder, I think a lot of people are embracing the outdoors as a way to get active, get social, but still be safe in the age of coronavirus. So today's presentation will focus on socio-ecological drivers of the black-legged tick in Maine. Kind of first started appearing in the 1980s, so really not that long ago. And in the last decade, we've seen a 5-fold increase in Lyme disease and other tick-borne diseases. So from both a recreational and medical standpoint, it is something that is of great interest and importance to the people of Maine and to the scientific community. So Ali, I will let you take it away.
All right. Well, thanks. Thanks so much, everyone, for joining us this evening, and thanks for the opportunity to speak with all of you. So I'm Allison Gardner. I'm an assistant professor in the School of Biology and Ecology at UMaine, and I've been here for about 4 years now, just beginning my 5th year. And I'm a medical entomologist. And actually, prior to coming to UMaine, much of my research has focused on mosquito-borne disease ecology and dynamics. But I was really excited to move to Maine and to shift some of my research emphasis in my lab to tick-borne disease over the past couple of years. Because as we all know, Lyme disease is arguably the most important vector-borne disease in North America. I grew up in New York, and honestly, it is harder for me to name people who haven't had some sort of personal experience with Lyme disease than people who have. My parents, my sister, and I all had Lyme disease at some point in the 90s and the early 2000s. And it really feels like the occurrence of human cases in new locations regularly is headline news. And this is really backed up by the data, right? So here we're looking at a map of the number of Lyme disease cases per state in 1992, which is the year that Lyme disease became a notifiable disease, you know, that doctors had to report to the CDC. And take a look at this next map. You know, here we are 25 years later in 2017. So the different colors on the map represent the states ranked by the number of reported cases. So darker green means that there are more reported cases, and the lighter colors represent fewer reported cases. And so I'm just going to switch back and forth again between these two maps so you can notice the change over time. First of all, you'll see that the number of reported cases of Lyme disease has multiplied by 4 over that 25-year time frame. You know, we had 10,000 roughly reported cases in 1992 compared to over 40,000 in 2017. And these numbers actually likely represent a 10-fold underestimate of the total number of human cases. So you can really see and appreciate how the public health burden of tick-borne disease has increased over time. And we also see a dramatic increase in comparing these maps in the number of cases in 2 major foci of transmission. One of them is in the upper Midwest, where I was working prior to coming to UMaine. We've got really dramatic increase in the number of cases in Wisconsin and Minnesota in particular. And then, of course, up here in the Northeast, we've really seen the number of cases of Lyme disease explode. You know, in Maine, we went from 16 reported cases in 1992 to over 1,800 in 2017. That's a 100-fold difference in the number of cases. And so I personally was really excited for the professional opportunity to move to Maine to study this problem. And that's a large part of what my lab's research has been focused on over the past few years. All right. And so I direct the vector biology lab at the University of Maine. And my students and I work on a variety of different research problems related to infectious disease that's transmitted by arthropods. But all of our efforts in the lab are thematically linked by three focal research aims. One of our first major goals is to investigate the interacting ecological and social conditions that drive arthropod-borne disease transmission and spread. A second goal that we have in the lab is to try to predict the spread of emerging and re-emerging arthropod-borne diseases in the landscape. And finally, our third broad goal is to translate our findings to devise effective disease control strategies that are grounded in ecological theory and offer sustainable long-range means to manage arthropod disease vectors. So throughout all of these projects, my lab and our collaborators use concepts and tools from a range of disciplines spanning entomology, which is the area of my training, community ecology, data science, molecular biology, so on and so forth, to understand how ecological processes drive arthropod-borne pathogen transmission and ultimately contribute to human health outcomes. And of course, front and center to what we're working on is this question of why Lyme disease is emerging in Maine right now. What are the ecological processes that are responsible for this trend? Where can we expect Lyme disease to go in Maine from here? And that's going to be the focus for my talk right now. I'm planning to spend the next 20 to 30 minutes or so giving a broad overview of some of my lab's research. And then I also am planning to leave plenty of time for questions. This is obviously a huge subject and really pertinent to the state right now. And I'm happy to discuss any questions that you might have on the subject. All right. Now, part of the reason why Lyme disease is so challenging to study and manage and forecast is the complex life cycle of the black-legged tick, which is the principal vector for Lyme disease here in the Northeast. The black-legged tick has a 2-year life cycle that you can see in the diagram here, with multiple molts from larva to nymph and then from nymph to adult. And at each molt, the ticks need to feed on a host, on a vertebrate typically. And so for the larvae and the nymphs, the hosts tend to be small mammals, and these also are many of the species that carry the Lyme disease bacterium, Borrelia burgdorferi. And it's important to note that once a tick feeds on a host that's infected with a parasite and it acquires that parasite, it's going to remain infected for its entire life cycle. And so if a larva feeds on a mouse that carried the Lyme disease pathogen and then takes 2 additional blood meals from other hosts in the nymph stage and in the adult stage, each of those subsequent feedings where the tick feeds on a vertebrate host is another opportunity to pass on the pathogen. All right. Now for adults, the hosts typically are larger mammals that may include deer or dogs or people. And although these hosts don't actually harbor pathogens, these ticks, these larger mammal hosts are not capable of actually passing on a pathogen to a tick. They also can serve to amplify tick-borne disease risk by supplying blood meals to large numbers of ticks and therefore increasing the tick population size. And so when we think about tick-borne disease risk in the environment, typically we're thinking of 2 different factors. First, we're thinking about the density of ticks in the environment, and then we're thinking about the pathogen infection prevalence, which are driven by different hosts and sometimes different ecological processes. Now we're going to add on to another layer of complexity. And when we think about ticks, this really is often what we think about first, these interactions between ticks and their hosts, because that's when disease transmission takes place. But the reality is that ticks spend over 90% of their life cycles off host, not attached to a host, not parasitizing a host. Instead, they're living on the forest floor, where their ecology and persistence in the environment is driven largely by abiotic conditions. So this is another view of the tick life cycle in Maine. you can see here that the ticks have, two major periods throughout their 2-year life cycle. We've got dormant periods where the tick is inactive, and this includes the nymphal and adult overwintering periods, as well as other parts of the life cycle where the tick is just digesting a blood meal after recently having fed on a host. And then we also have questing periods, which are periods in the tick life cycle where the tick is mobile and actively seeking a blood meal host. So these are all parts of the tick life cycle that are spent not attached to the host. And the survival of ticks through these periods where the tick is off host are driven by a variety of different environmental factors, mostly related to microclimate conditions. During periods where the tick is dormant, the tick population and tick survival might be limited by temperature, be it low temperature in the winter or high temperature in the summer, also by snow cover in the winter, relative humidity. In general, a low relative humidity predicts a lower tick survival rate, as well as variation in weather. If we have periods of like extreme variation in temperature, precipitation, or relative humidity, that also could be very difficult from an energetic perspective for the tick to withstand. And then during the questing periods where the tick is actually seeking a host, ticks could be limited by temperature, again, highs in the summer, cold springs or falls, relative humidity, again, variable weather conditions, and the duration of timing of questing activity. So the reason I'm talking about all of this is because I want to illustrate to you, what makes it so challenging to study and predict where and when the tick is going to invade a new location, and some of these different variables that we need to consider when it comes to predicting the emergence of tick-borne disease and understanding the current distribution of tick-borne disease in the state. And so this talk is going to be one part about tick biology and one part about the scientific process and how we go about dealing with such a challenging system to study. And the complexity of the tick life cycle is in fact one of many challenges to studying this complex problem of where, when, and how ticks are spreading throughout Maine. One of the major challenges that we're dealing with when we conduct research related to tick ecology and invasion ecology is the simple reality of tick detection probabilities. Well, I show here in this top picture, ticks can be the size of a poppy seed. This is an image that the CDC uses often in their outreach, simply to illustrate how small ticks are when you're searching your own body for ticks after spending time outdoors. But the same issue comes into play when we're searching for ticks out in the field in a research setting. How do you study a process that you essentially can't see? These ticks are just so small, very hard to find them. Another challenge when you're conducting this sort of research related to invasive species and geographic range expansion is prioritizing which types of data to collect. As we saw, there's really a wide range of biotic and abiotic variables that drive the tick life cycle and the likelihood of ticks surviving to feed on hosts. And so it's challenging to sort out, given finite resources, which types of data are you going to collect? Similarly, when we're conducting long-term research, trying to understand the invasion ecology of ticks, it's challenging to anticipate the future data needs. What we think is a problem of interest right now might not necessarily be the same problem that we're interested in years from now. And so as we think about long-term research concerning invasive species, anticipating the type of information that we might want to know in the future is another challenge. There's the challenge of mechanistic inference across scales of space and organization. Chances are there are very different processes that are driving the survival of ticks when individual ticks versus the movement of ticks at the population level and at the landscape scale. There's also the challenge of how to get this research funded to begin with. Selling research to funding agencies that have a range of different priorities. Some funding agencies will be more interested in basic phenomenological research. Other agencies are more interested in application. And so figuring out how to fit this long-term research that's needed to understand the invasion ecology of the tick is a problem in itself. as well as the logistics of conducting long-term research. And so what I think all of this serves to show is that there's really a need for a multi-pronged approach when it comes to studying the emergence of tick-borne disease and why it's occurring now. This is not a problem that we can really understand from one type of data alone. We really need to integrate different types of data streams to be able to understand across different scales what ecological processes are driving the invasion of the black-legged tick and the emergence of Lyme disease in Maine. And so my goal for the remainder of this time that I have with you is to give you a broad overview of how my lab is going about tackling these problems. And again, then I'll leave time at the end for your questions and thoughts. So one of the primary tools that we initially use in trying to understand the ecological drivers of the occurrence of ticks is conducting detailed case studies of specific locations of interest. And so the advantage of this type of study is that you can get a lot of very detailed information about the landscape scale drivers of tick habitat use and pathogen infection prevalence through intensive sampling within a small geographic area. And so a lot of our research actually has been focused on MDI in Acadia National Park, which is an appealing location for a couple reasons. First, it's a highly visited and epidemiologically relevant part of the state where there are lots of people who might become exposed to tick-borne disease. And then second, another logistical advantage of working on MDI is that there are really diverse landscape features within a relatively small geographic area. And so one of my initial research interests upon moving to Maine was how forest habitat might constrain or not the distribution of ticks, because Maine is characterized by so many different forest types compared to New York and southern New England, where the black-legged tick has been more extensively studied. And so just to walk you through very briefly what our case study in Acadia looked like, over a period of the past 2 years, my lab has been collecting and counting ticks off host at different sites spaced throughout different habitats in Acadia National Park. Then we've been examining statistical associations between tick counts and habitat features at broad scales, like land classification and elevation. And then ultimately, one of the goals of our research has been to model the distribution of ticks on MDI based on these habitat features. So the darker red represents areas on MDI that have a much higher probability of tick presence based on our field data collection. The areas in blue have a much lower probability of encountering ticks in those areas on the island. All right, so case studies really is a major component of the research that we've been conducting. But the problem is case studies can't really explain ecological mechanism. We get a good sense of distribution of ticks and some of the landscape scale drivers of tick occurrence from these case studies, but we don't really know why those landscape features are driving the occurrence of ticks. And so another facet to my research program that's actually been a major component of some of my early research in Maine is field experiments, where we actually conduct some sort of manipulative effort to understand the processes that are driving where you find ticks and where you find pathogens. So one example of a field experiment that my lab has been running over the past couple of years is focused on how forest management practices may influence tick-borne disease transmission dynamics, and even whether forest management could inhibit tick-borne disease exposure risk at the property and landscape scales. And so, forest management actually is a very appealing tactic for tick-borne disease management because it has the potential to affect multiple stages of the life cycle. And regardless of whether our primary management approach is, prescribed burns or invasive plant removal or logging, we know that forest management practices can influence both microhabitat features like temperature and humidity and leaf litter cover that in turn determine what proportion of ticks survive the off-host portion of their life cycle, so directly leading to tick abundance. And we also know that management practices stand to impact wildlife communities. So factors like vertebrate host diversity, white-footed mouse density, one of our principal pathogen reservoirs for tick-borne disease, white-tailed deer density can impact both the abundance of ticks in the landscape as well as tick-borne disease prevalence, collectively leading to changes in tick-borne disease exposure risk as a consequence of land use. So we've been conducting a field experiment over the past couple of years focused on the impacts of timber harvesting on tick-borne disease ecology. So one of my former master's students, Christine Conti, recently completed her thesis project in my lab, which focused on tick densities and pathogen infection prevalence in forest stands that had recently been thinned within the past 5 years versus no harvest history within the past 20 years. And what Christine found in her research is that black-legged tick density generally is lower in stands that have been harvested recently compared to those that don't have any sort of recent harvest history. And again, the virtue of these field experiments is that we're able to get a much better handle on some of the mechanistic drivers of these patterns compared to some of like the broader scale landscape studies that we previously talked about in the case studies. And in the case of Christine's research, she found that microclimate conditions is one of the variables that seems really strongly related, a potential explanatory mechanism to explain this relationship between tick density and timber harvesting. Overall, she found that using a variety of different temperature and humidity data loggers, she found that temperatures tend to be higher and humidity tends to be lower in more recently harvested stands, which it makes sense when you think about it. If you've got canopy gaps and sunlight exposure on the forest floor, that's going to reduce the probability that ticks are able to survive under those conditions. Another mechanism that Christine studied that I think could also explain this pattern that she observed is host availability. Christine had also been conducting live trapping of small mammals in these different stands that she was working in, and she found that overall the species, the abundance of rodents was lower in the harvested stands compared to the control stands, and overall the species diversity in the harvested stands also was higher than in the control stands that tended to be dominated by white-footed mice. Now here's another example of a field experiment. Another area that my lab has been interested in studying is tick survival over winter, because conventional wisdom is that winter temperatures impose a major constraint on the geographic distribution of ticks in the state. So this is another question that we've been, another piece of the puzzle that we've been examining using an experimental approach. Another master's student who recently graduated from my lab, Michelle Volk, conducted a study of tick overwinter survival spanning large-scale ambient temperature and snowfall gradients in Maine. So she had these 5 field sites throughout the state, and at each field site, she established 16 enclosures that look like this. She seeded them all with live ticks, so there's live ticks in these buckets underground. And then to each enclosure, she applied one of 4 experimental manipulations. For some of these buckets, she removed snow and leaf litter that would provide some protection to the ticks over winter. For some, she removed the snow but left the leaf litter. Then the opposite, leave the snow and remove the leaves. And then she had a control treatment, probably the most ecologically realistic treatment, with both snow and leaf litter present. And what she found in the context of this study is that under both cold ambient conditions and comparatively warm ambient conditions, just to show you some data here from the 2 extreme ends of this climate gradient, snowpack and leaf litter really serve to enhance overwinter survival of black-legged tick nymphs. Even in Presque Isle, very far north, very cold conditions, the ticks were able to survive. Over 60% of the ticks were able to survive despite the cold ambient temperature if they were insulated by leaf litter and snowpack. And so this is another field experiment that demonstrates a valuable mechanistic understanding of how overwinter climate could potentially be constraining or not the distribution of black-legged ticks. All right, so we've been conducting these field experiments. Of course, another, all these approaches that we've discussed so far are good for understanding fine-scale patterns of tick occurrence and perhaps even some of the causal ecological mechanisms that explain why these patterns occur. But if our goal is to study the emergence of ticks in Maine, we also need to get to larger spatial scales. And so my lab has been running 2 projects involving active surveillance, where we actually go out and search for ticks in the landscape to monitor the invasion of black-legged ticks. And so the advantage of active surveillance is that you do get consistent data collection that enables inter-year comparisons of where the tick is spreading from year to year. And in theory, we could document the invasion of the tick in real time. The downside to active surveillance, of course, is that it's very laborious. And so my lab has been conducting, again, using the same sites I highlighted in the previous slide, spanning the climate gradients in the state. For the past couple of years, we've been conducting off-host tick surveillance every 2 weeks from June to August in each of these 5 locations, some of which include areas where Lyme disease incidence currently is very high, some where Lyme disease incidence is very low, and then some locations where Lyme disease effectively is absent at this point, and we don't expect to see very high tick densities. And so our ultimate goal here is to be able to track the invasion of the black-legged tick into these new locations and understand the processes that seem to drive the spread of the tick. So again, we've been conducting this off-host tick collection. We've been live trapping small mammals in August in order to estimate small mammal population size and tick burdens and how that might relate to the invasion ecology of the black-legged tick. Conduct scat surveys in May to estimate deer activity. And we test a certain amount of ticks and small mammal ear biopsies per year for pathogens as well to monitor the concurrent invasion of tick-borne pathogens throughout the state. We're also monitoring temperature and humidity, rainfall, snowfall, and how all of these variables are associated with the occurrence of the tick. So again, really laborious process. We're also engaged in a second active surveillance project, the Maine Forest Tick Survey. And this project is actually a citizen science project. And so the goal here is by effectively teaching other people how to collect ticks on their own properties, we'll be able to reach we have the advantages of active surveillance where we're collecting ticks using a standardized method that enables comparisons of tick densities across locations and across years. But we're able to reach a wider array of field sites beyond like those 5 field sites where we've been doing a lot of really intensive monitoring within the lab, by getting people to collect ticks on their own land. And so this past summer, 2020, was our first summer of the Maine Forest Tick Survey. We enrolled 125 citizen science participants who collected ticks on their own properties. We're going to be enrolling the same number of new participants next summer as well. And all of the citizen scientists who participated in this work were located in southern and coastal Maine, which is our pilot area where we're trialing out this project. They all own woodlots that are greater than 10 acres. And so the participants in this active surveillance study drive for ticks on their own land. They complete questionnaires about their forest properties, and then we test the ticks for pathogens back at UMaine. Now, passive surveillance is another approach that we're using to understand the invasion ecology of the black-legged tick. The state of Maine has actually offered tick identification and pathogen testing services for 30 years. So this is not work that's being done in my lab, although I do collaborate with these labs. The Maine Medical Center Research Institute led the tick identification service from 1990 to 2013, where people can mail in a tick that they collect on themselves or their pets. They'll get the tick identified. UMaine Cooperative Extension assumed control over the passive surveillance program starting in 2014 to present. So passive surveillance is a little different from active surveillance in that ticks aren't being collected using a standardized method. We can't really get estimates of tick densities or and that there's not really a systematic way that ticks are being collected. However, passive surveillance is valuable alongside some of these other tools because it offers a means to detect the arrival of ticks in new locations and document tick occurrence patterns over time. And so we've been conducting a range of research efforts also using passive surveillance data. This is a study that I just published with Susan Elias, who works at MMCRI. in the Journal of Medical Entomology, where Susan was able to use the passive surveillance data from 1990 to 2013 to document associations between winter temperature and deer densities and the occurrence of ticks through this active surveillance data. Griffin Dill is a PhD student in my lab currently, who's heading up the tick identification and pathogen testing service at UMaine Cooperative Extension now. And so these are, you can see here, these are the data, the number of ticks that were submitted in 2019 through the passive surveillance program. You can appreciate there's much wider coverage that you get from these data compared to any other form of active surveillance. So as part of his thesis research, Griffin is working on developing species distribution models for the black-legged tick based on these passive surveillance data. And then the final tool in our arsenal to understand the invasion ecology of tick-borne pathogens is epidemiological data analysis. Just to give you a flavor for what our epi data in the state look like, on the y-axis, we're looking at the incidence of Lyme disease per 100,000 people over time. On the x-axis, we're looking at the year, and we're also going to bring some maps into play. You can see here that over time, the incidence of Lyme disease is really increasing dramatically, as is the distribution of Lyme disease cases in the state. Now, there are some limitations on use of epidemiological data to understand the distribution of ticks and pathogens, because you don't know where the person who became ill actually encountered the tick. All of the human case data are reported based on the person's home address rather than the place where they likely encountered the tick. So maybe they got bit by a tick in their backyard, maybe they drove down to Massachusetts and got a tick bite there, became sick, and then the case was reported in Fort Kent. We have no idea to know. We have no way to know. But these maps show that the number of black-legged ticks submission to the state's passive surveillance program between 1989 and 2000, to the left, 2001 to 2013 on the right, you can really appreciate the difference, and that the distribution of the tick and the distribution of cases of Lyme disease over this time period are very similar. And so I think that human case data also can offer a reasonable proxy for tick occurrence data to study the range expansion of ticks, because in general, human cases are far more likely to be reported by doctors than ticks are to be submitted by individuals to passive surveillance programs. So we are able to access data related to the emergence of Lyme disease that has this much greater coverage and much richer data source through the human case data compared through any tick data that we collect. So I want to wrap up just kind of by going back and summarizing the different approaches that we've been looking at, and some of their strengths and limitations. So again, the case studies that we looked at first, they do offer this very detailed understanding of a specific location, and it's possible to investigate mechanistic drivers of why ticks occur where they do, but it may not be generalizable to other locations. The field experiments, on the other hand, I think are the gold standard, the best way to examine the mechanistic drivers of tick distribution, but they may not translate easily to larger spatial scales, and they also can be quite expensive to carry out. The active surveillance is really useful in that it enables consistent data collection and inter-year comparisons. You also can use active surveillance to document the tick invasion in real time. But on the flip side, it's extremely laborious and time-consuming. It might only be possible to visit a limited number of locations. And active surveillance also, depending on how it's conducted, may or may not lead to a mechanistic understanding and explanation of why ticks occur where they do. The passive surveillance offer the strong ability to detect the initial introduction of the tick to a new location, and it also is relatively inexpensive. However, with passive surveillance, we can't study tick densities. We're looking at presence-only data, so we don't have any records of where ticks are absent. And there also may be biases in data coverage depending on how aware people are of the existence of the passive surveillance program as an option. Epidemiological data offers consistent reporting over a large geographic area, but again, no indication of where the individual encountered the infected tick. So we've got a really wide range of different approaches here. And moving ahead, we know that the black-legged tick and actually multiple tick-borne pathogens currently are diffusing throughout Maine. The range expansion of the ticks and the pathogens likely are driven by a range of ecological processes, different scales of space and biological organization. And so I think it's really important to use this blended approach involving some combination of active surveillance, passive surveillance, epidemiological modeling, and then these fine-scale field studies and experiments in order to illuminate the ecological drivers of the tick's range expansion across scales. There is a major conceptual challenge that exists here involving how to actually assimilate these different data streams into predictive models, since as you've seen, the data collection approaches are quite different across these different techniques, and the types of insight that they offer are quite different as well. But on balance, I think that there's real potential for an improved mechanistic understanding of the range expansion of ticks and a real value of it to inform public health practice, as well as a framework to study future invasions of other arthropod vector species. And so, so I'll just wrap up my portion of this by acknowledging members of my lab, past and present, as well as collaborators around the state who have been involved in this research with me. I leave my contact information up here as well in case you've got any questions you'd like to talk about following this session this evening. And now I'd just like to open everything up to you. Happy to take any questions and about anything tick-borne disease related in Maine. Thanks.
Wonderful. Well, thank you, Allison. That was enlightening, fascinating, potentially going to make me more vigilant when I'm out and about. So firstly, I want to say thank you to you and your lab for doing this important work. Having seen the population and the incidence of Lyme disease and other tick-borne diseases increase so drastically, it's comforting to know that someone is at least trying to track what's happening so that we can make smart decisions for the state and for ourselves. So the first question you've kind of already answered, but the first question I usually ask our presenters is, what's next with your research?
Yeah, so it's really the very beginning of the research program here. We're only 4 years in, but yeah, I really think that we've got all of these different data streams that we're considering at this point to try to understand what's driving the range expansion of ticks. We've got data sources that range like the really local level and studies of small numbers of ticks and the ecology and physiology of individual ticks, ranging up to these landscape scale studies where we're trying to understand how ticks are moving in the landscape and what processes are driving the spread of ticks in the landscape. And so certainly one of our major goals is to continue this long-term research, observing the expansion of the ticks in real time.
Wonderful. So the next question comes from Margie, and she is asking on behalf of her mum in Connecticut, who has had Lyme disease more than 30 times.
Oh, goodness.
We know that Lyme is not great, and of course if you don't catch it, it can have long-lasting effects. So Margie's asking, can feeding birds in your yard be a problem in that they attract mice and chipmunks that can carry the disease? And she adds that her mum is a gardener, so obviously spends a lot of time outside, but loves to feed the birds, which of course brings with it the added extras that are not meant to be getting the bird seed, but do.
Yeah, so this one I've heard mixed advice about. I haven't seen anything published in the way of scientific articles about the impacts of bird feeders on the likelihood of encountering ticks. The line of logic here is that if birdseed gets on the ground, especially like outside the winter months, that this is going to provide a food resource for mice and draw more mice into your yard. And then that could increase the density of ticks in your yard. My personal inclination is that feeding the birds is far less of a problem compared to leaf litter in the backyard. If I were to advocate for one particular management strategy that you could do on your own property, it would really be to rake the leaves and keep out of the leaves. I think that the leaf litter we've seen provides critical habitat for the ticks, including over winter. So more leaf litter enables the ticks a much higher chance of surviving over the winter. I personally believe that Perhaps it's true that that the bird seed on the ground could provide some food for mice. But as someone else who loves feeding birds and who derives a great deal of joy from it, I would, if I were approaching management actions on my own property, which I do, I think raking leaves is a lot more important than like denying yourself the pleasure of seeing the birds because of some extra food that might go toward the mice.
Okay, that's good to know, because I too love feeding the birds, and especially in winter when
Especially in the winter.
Everything kind of goes to bed. It's nice to have a little bit of life in your garden. So
it is.
The next question is kind of about you personally, and how has your knowledge of the prevalence of ticks and Lyme disease influenced how you spend your time outdoors?
That's a great question. Well, I will say that that no one in my lab has ever gotten Lyme disease or any tick-borne disease while actually out conducting research. I have gotten Lyme disease when I have, not at work, off the clock. And so I think that probably speaks to the fact that I've been maybe a little too lax at certain points about protecting myself against tick-borne disease. The main way that I protect myself against tick-borne disease when I'm out like hiking and gardening is by trying to be really thorough with the tick checks. I think that that's really the best way to protect yourself. I personally don't really like using insect repellents, and as much as it probably is the best strategy, I don't wear long pants and tuck my pants into my socks when I'm out hiking. It's impractical. I think that the best way that you can protect yourself is just by being really aware of the ticks. Whenever I get back into the car after hiking or whenever I get home, I'm always checking around my legs. There are certain areas of the body where you're more likely to have ticks attached than others. So I'm always checking my legs, I check my back, I try to check areas of the body that can be a little hard to search for ticks and remove them as soon as possible. So I think that really the best defense is knowing how to remove ticks, always being vigilant after you return from hiking. And those are really the main approaches that I use to protect myself when I'm out hiking.
Great. Well, hopefully now everyone else will adopt that. I know that I'm very vigilant for my son, but for myself, I'm probably not as careful as I should be. The next one comes from Hannah, who's an undergraduate student at Maine, Machias, and she's thinking about conducting surveillance on black-legged ticks for her senior thesis. Seems like her goal is to kind of do a similar thing to what you're doing throughout Washington County. Do you have any quick advice or recommendations for her?
Well, that's great. If you send me an email, I would love to talk to you about your project. We have done some monitoring for ticks in Washington County, but not really extensive. We've basically been confined to color. Washington County is so important because that's really like the boundary of where we see human cases of Lyme disease. So it'll be really important to know, when ticks start showing up in Washington County and what sort of habitats are facilitating the ticks. The main way that we collect ticks off host is by using like 1 meter square, white flannel and just dragging it on the ground. The ticks are typically the way they search for a host is by standing in the vegetation and kind of like waving their legs in the air, and they just wait for a vertebrate host to pass by and latch on. And so when we collect ticks, the idea is that the flannel cloth mimics the vertebrate host. The ticks will attach to it just as soon as they'll attach to a vertebrate, thinking that flannel cloth is a host. So I think that that's like a really nice, straightforward way of collecting the ticks. It also has the advantage of being kind of fun because you spend a lot of time just walking around in the woods dragging this cloth on the ground. I find this a very meditative process. But yeah, I'd be delighted to talk to you more about your project if you have an interest. I think that understanding what's going on in Washington County is really interesting and really important because that is where the ticks are only just starting to arrive.
Great, I was about to say, and it sounds like dragging for ticks is the therapy we didn't know we needed.
I think the citizen scientists have been learning that this summer.
So the next question comes from Stephen. He asks, what are the redeeming social values of ticks? Like, do they have a positive role to play?
That's a good question. I was not asked this question about ticks, but I was asked this question about mosquitoes in my comprehensive exam in graduate school. And the answer that I had to give was, I would kill them all. I suppose that in the context of the mosquitoes that they might have a certain niche in the food webs, but honestly, it strikes me as a niche that any number of small flies could be able to satisfy. I think that when you trade off the whatever limited benefits the disease vectors might have to the ecosystem, to my mind, the detriment to public health is just far overpowers any benefit. For the ticks, man, I cannot think of something that I view as a redeeming quality. They don't seem to be a major food source for any animals. There are like some animals that feed on ticks, mostly other invertebrates. Invertebrates are the major top predators of ticks. I am hard pressed to think of something redeeming about this animal.
Well, it's good to know that the science perhaps backs up how most of us feel about ticks in the first place. So thanks for that. The next question we've got is there any evidence that the rate of transmission of Lyme or other tick-borne diseases does or doesn't necessarily parallel tick number? So I guess as the population is growing and the rate of Lyme disease incidence has grown, are they rising in parallel, or is there a different ratio?
Okay, so if I understand the question correctly, the question is like, are tick densities and Lyme disease incidence really like increasing in parallel to each other?
Yeah. Yes.
And so this is a problem that I've actually been just starting to work on in Maine. But in the Midwest, where I've worked previously, I conducted a study where we were analyzing at the county scale the occurrence of the tick and the incidence of Lyme disease before and after the tick first arrived in a new county. And we found a very strong association there, where after the tick was first introduced to a county, the tick-borne disease incidence in humans would increase 5-fold, which I think is really dramatic. And so I strongly suspect, part of our interest in these long-term studies of ticks and tick-borne pathogens in the state is to understand that exact question concerning the sequence of the arrival of ticks and the arrival of pathogens and the consequences for spillover into the human population. But I strongly suspect that ticks and pathogens are arriving to new areas roughly simultaneously, and the arrival of the tick is what triggers this large-scale spillover that we're seeing into the human population. It is an important question. Right now I'm approaching it using a combination of like the passive surveillance data as well as the epidemiological data as well. I think that there's some nice study designs where you can analyze those data in parallel with each other to get at that exact question. But based on what I've seen in the Midwest, I strongly suspect that there's a good association between arrival of the tick to a new location and increase in the incidence rate in humans.
Interesting. I guess now this question kind of goes a little bit in line with the one I just asked just before. In addition to Lyme disease, what other diseases can be carried by ticks? Obviously, Lyme is the one everyone knows, but I know there are a number of other parasitic and tick-borne diseases that you can.
There are, yeah. So in Maine, really the big three that we focus on are Lyme disease, and then human anaplasmosis and human babesiosis. Powassan virus also got a lot of press a few years ago when there were a few human cases in Maine. And so these are all pathogens that are transmitted by the black-legged tick in particular. And in terms of the distribution of these pathogens, they're all spreading. Anaplasma probably has the second largest geographic distribution after Lyme, and then Babesia has a smaller geographic distribution. It's more localized to southern and coastal Maine right now. But all three of these pathogens are spreading. And this is honestly why I believe that understanding the invasion ecology of the vector and of these pathogens is so important. Even if we were to get a vaccine for Lyme disease, for instance, we still have all these other pathogens that are on the rise that are being transmitted by the black-legged tick. And really the only effective way to manage all of those pathogens simultaneously is by managing the vector. We also have other tick disease vectors that occur elsewhere in the country. The Lone Star tick you might have heard of, that's the one that causes the red meat allergy. It also is a vector for ehrlichiosis and a few other tick-borne pathogens as well. But Lone Star tick there's no evidence of an established reproducing population in Maine yet. So it's probably, another 5-10 years out before we're dealing with that as a serious problem. And then on the West Coast, there are some other ticks, tick-borne pathogens, Rocky Mountain spotted fever. Again, not such a huge problem in the Northeast, but certainly elsewhere in the country.
Great. I've got just a couple more, and we've got a few more minutes, so hopefully we can get through them all. Someone has asked, is it okay to put wood chips on your paths, or does that increase tick numbers? So I can imagine beautiful gardens where all the leaf litter has been removed, but then if you add wood chips to your path, are you just giving the ticks another place to winter over?
So that's a good question. I haven't seen any data concerning this. Usually when I see ticks in those habitats, this is purely anecdotal. It's dog ticks. So one of the key things, that I think is worthwhile to learn is how to differentiate between black-legged ticks, which are disease vectors, and dog ticks, which really are not disease vectors in the Northeast so much. And whenever I see ticks really very close to my home, it's usually dog ticks, which still no one likes the idea of having a tick attached to them, but at least they're less likely to transmit disease. They're very easy to tell apart, actually. If you look at pictures of dog ticks and black-legged ticks online, you can see that they have some very distinct features. Black-legged tick most notably has black legs, dog tick does not. And so, yeah, I think I can't give a 100% answer about the wood chips in particular. I suspect that the leaf litter is by far the more substantial problem when it comes to property scale management.
Okay. Thank you. Well, I feel like I now have a convenient excuse to not put wood chips down because I need to focus on getting rid of my leaf litter instead. So the next, I guess it's a question as well as a statement. Do you know if the tick lab has considered incorporating veterinary clinics for submission of ticks? Now, I know the tick lab will do a free ID, and I know you can submit it for testing for a small fee. If people are concerned about their pets, can they supply a tick to the lab and have it tested?
That's a good question, and I do not know the answer to that. I'm sorry. I'll need to point you to Gryffindor. Yeah.
That's quite all right. We don't expect you to know everything. So Tegwin, an answer to that is possibly you can get in touch with the tick lab. I put the link to it in the chat box earlier, and I know that basically goes through all the options that you have there. It's a very interesting thought, though. And then this is another one that's probably a little bit outside your field, but you may kind of keep an eye on it. Someone has asked, where are we at with the Lyme vaccine?
Yeah, so that's another one that's really outside my scope as an entomologist. There have been various challenges to developing the Lyme vaccine over the years, and honestly, diagnosis of Lyme disease in humans also is a minefield. I don't think that we really have any perfect diagnostic technologies either, which is part of the reason why Lyme disease has become such a great medical controversy. The vaccine has certainly been a priority of funding agencies, especially Department of Defense, for years. Same for several other vector-borne disease vaccines like dengue in the case of mosquito-borne disease, Zika. But it is a challenging process to develop the vaccine, for the bacterial disease and for tick-borne parasites. And so I don't know exactly, what the outlook is, but I continue to maintain that I believe that vector management and our personal behaviors are the best way to confront tick-borne disease, especially because there's so many other pathogens that we now know these same vector tick species are able to transmit. And thanks for the question.
Thank you. And then one last one. I'm just going to make sure I've read it correctly. Someone has asked, do you know if the disease can be transmitted, what's the word, transovually. So
That's a great question.
Pathogen to organism to its offspring by infection of eggs in its ovary.
That's a great question. And the answer is no. There is no evidence in the field of transovarial transmission of Borrelia burgdorferi. And so that means that, that ticks only are able to acquire the pathogen from a vertebrate host. No larvae are going to have a pathogen. They can't be born infected. And so, you know, which host species the larvae are feeding on is really consequential in terms of the likelihood of the tick acquiring a pathogen.
Well, I guess that's some good news. Maybe that's the redeeming factor of the tick, is that.
Which we can't say for the mosquitoes. Lots of transovarial transmission of mosquito-borne disease.
Well, I think that brings us to the end of our questions. Some wonderful questions out there. So thank you so much for asking such thoughtful questions. And Allison, again, thank you for your answers. Thank you again to you and your team for the work that you do in this important area that is of, I guess, utmost imperative for Maine as we see these populations grow. So I just want to say thank you for joining us. Everyone else, thank you for joining us too. And I know it's hard to believe, but again, this is the last cafe of the year. So Allison, you win the special crown of the last 2020 Science Cafe. And we will be restarting in January, so keep an eye on your emails for when we start up again, and we'll bring you some more interesting topics next year. But for now, I just want to say thank you. I hope you enjoyed. And Allison, thank you again for joining us tonight. It was fascinating.
Well, thanks so much for the opportunity.